There we go. Yeah, the menu was hidden. All right, now we're set.
Okay,
All right. Yay.
so I was tasked with talking about general chem cure ideas and syllabus with a focus on drug design, which is something I've been doing for a couple of years now. And so if I can have my next slide, please. I always like to show this when I'm talking about cures, because many times, however many times you've taught a cure, even if it's the same cure over and over again, you always feel as though you're working at the edge of chaos. And the job is to manage the chaos, not to eliminate the chaos, because the chaos is one of the things that contributes to actually the student outcomes. So next slide, please. We'll go quickly through a couple of these. This is, and you've seen this several times now, but the elements of a cure, or in this case, the elements of research. And the only thing that's on this one that isn't on the other one is research ethics, but it's got the same elements of relevance and all the other things you've heard about. So next slide, please. I want to point you to a resource that might be useful. This is a ACS publication that ACS asked Betsy and I to pull together, and you'll see from the author list that many of the people are MCC members, but it goes through all of the different aspects of a cure. And if your library doesn't actually have this and you want a particular chapter, please feel free to email me. And I put my email on this, and I will send you a copy of a chapter, with the condition that you don't circulate it freely. It's for your own personal use. Okay, next slide, please. Okay, so once you've decided you're going to teach a cure, and we're going to talk about um, the cure, I've been teaching this introduction to chemical thinking through the lens of anti-malarial drug design. That's the one, and the CureNet links to the CureNet here, and there's a whole bunch of resources, and I'll let you look at those in the interest of time offline later. So if you can go to the next. So one of the nice things about these cures, and Chris talked a little bit about computational resources and approaches, you can incorporate a lot of computational chemistry, biochemistry into it. The advantage of that is they're free. The second advantage is that they can often be used asynchronously out of class, and you can pair them up with the appropriate wet lab experiments. So it's a way of sort of keeping your budget down if you've got a limited budget, but it's also a way of making use of state-of-the-art computational cures. And the link there just takes you to some of the computational resources. So don't bother to click on it now, but you can explore them from that. So if you go to the next slide, please, Joe.
Oh, oh, too fast. There we go.
Okay, so you basically need some teaching approaches to help students develop their hypotheses. And again, as you'll see when you get into the detail of some of the links here, we put a lot of resources up on the web of ways to do this, and PowerPoints. We published some papers about how to develop a hypothesis, etc. But you need, ways to do that. You need ways to allow students to repeat important experiments. I saw there'd been a discussion on Slack about what repetition means and reiteration means. And you need all the typical things you've heard about already, so I won't dwell on these. But the nice thing is you can incorporate peer review into each and every stage of this if you plan appropriately. So next, please. And this now gets into the sort of project areas. And for this anti-pathogen drug design, you can do a chemistry focus introductory class, you can do a biology focus introductory class, or you can do an interdisciplinary introductory class. You can do it either modular, where you use about, maybe 6 weeks out of a 14-week semester, or you can do a full semester, just depending upon what they are. Now, tomorrow I'm going to talk a little bit about upper level classes and project areas, but any of these levels, there's a whole list of techniques that you can incorporate into them. You don't have to incorporate all of them, but depending upon what your department wants you to do, which if we go to the next slide, please. This just gives you now again that first link, our CureNet page for this particular anti-malarial drug design. You really should explore that because it's got all the details and lots of links and whatever. But there are two other links there. And again, I'm not going to spend any time clicking on them in the interest of time. But I want to get to the bottom half of that slide where it says pick a pathogen. Because it turns out you can use this sort of generic anti-pathogen drug design with almost any of the available pathogens that we've got. And we've developed some of them, and I think the T. brucei is through MCC, but we've got all of the ones that are listed there. So we've got T. brucei, which is African sleeping sickness. We've got T. krusei, which is Chagas syndrome, which is a dry mouth syndrome. N. fowleri, which is primary amoebic meningoencephalitis, otherwise known as brain-eating amoeba. And we're in the process of developing ones. If any of you are in Texas, you may have heard of the flesh-eating screwworms that are around at the moment and are probably going to cost billions of dollars to eradicate. And so we've got one we're developing now with that as well. We've got C. albicans, which is candidiasis, which is an oral cavity condition, and P. gingivalis, which is chronic periodontitis, or otherwise known as gingivitis. We've got all of those enzymes available, and if you're interested in collaborating on any of those, incorporating them into a GChem cure, let us know. Either email me or Jessica, and our email addresses are there. And those can be used in conjunction with the appropriate human isoforms, which you can get from Adgene, or you can con Joe into sending them to you. Okay, if we can move on to the next slide quickly. So typically, and this is one from the California Community Colleges Association, they've released what must be in, and you usually, especially with GChem course or introductory level courses, you're usually faced with some list of things that you have to include, and you can look at the appropriate list. This is one. There's another one on the next slide from USD. If I can go to the next slide, please. And you get a list of designated learning outcomes. And again, I'm not going to go through them one by one, but the next slide, please. And this is the particular ones that USD had for GChem requirements. And you had to fulfill in your course all 8 of those things to get it approved for GChem. Okay, we can go to the next slide. This is now a modular generic calendar to show how you can sort of lay this out and fulfill all of the areas in that and the sorts of experiments. So, this focuses on the various techniques. There's some computational, there's experimental techniques, there's spectroscopy, there's standard curves, there's pH titrations, all the things that we were required to put in there in this modular cure. And you can see it took up about six or so weeks. and then a final presentation at the end of the semester. And then I've given you in this some sort of way I broke down the points for the whole semester and why we handle this. These are all documented in the various sites I've talked about. So the next slide shows you the same sort of thing for a full semester cure. And this one really was fun because we developed some cool things for drug development ideas that were focused around what are called Lipinski's rules. If you don't know anything about drug development, there's some stuff in here. But there are these things called Lipinski's rules, and it entails looking at whether or not the potential compounds can penetrate a cell membrane, get through a membrane to get to the site. And we did a cool experiment with acid-base titration and phase separation, which fit right into the requirements for Chem and made the organic chemists very happy because it meant students were coming in knowing what a separatory funnel was and knowing what phase separation was all about. And again, the website has got templates for all of the experiments, and it's got rubrics for grading and those sorts of things as well. Then if we can go to the next slide, this is just the end of that. I stole from Dr. Jessica Bell this idea of doing badges in the class. So the students had to demonstrate that they could make up a solution, that they could work out the concentration of a solution they were provided with. They could make a buffer. They could set up a titration and do all the calculations. And they could use this thing, a measurement using what I call totals, a totals approach. And I found this was actually the most useful thing you can have in a cure, because most of the time students will ask you, how much should I use? Or things like that. And the totals approach, total stands for try, the T stands for try something, O stands for observe what happens, second T stands for think, the A stands for adjust and try again, and then the L stands for leave lab, smiling and satisfied, because they've worked out how to solve their problem and troubleshoot and think. And then there's a couple of examples on the next two slides of this totals approach. So next slide, please. So that's just a summary again of what it stands for with some pretty colors on there. So next slide, please. So you will inevitably, and we're going to do this one when you come to San Diego for the in-person workshop, the first question you always have is, how much enzyme should I use? And it's the perfect opportunity to introduce this totals approach. So, the picture shows you different scenarios, but you try something, the one you want to get is labeled D. And if you get A or C, you put in too much enzyme. If you get B, you put in too little enzyme. So you adjust appropriately and try again. And sooner or later, you get the perfect amount of enzyme. And you're extremely happy at this point because you get great data and you leave lab smiling. So the next slide's a couple of other scenarios. You can do the same sort of thing with establishing how much of a potential inhibitor to use, or to determine the concentration of a protein using Bradford dye and a standard curve, or determining the concentration of an NADH solution using Beer's law. They're all things where you can use this totals approach. So next slide, please. So this is a preview of some of the other things that I'm going to talk about tomorrow afternoon. We've talked about the drug design stuff. You could also do things with structure-function relationships, like the C-terminal phosphomimics and things like that, or other aspects of structure function. At the other, at the upper level, there's a whole bunch of cool things. And, you can look at allosteric regulation, substrate specificity, cofactor specificity versus carboxylic acid, the reaction mechanism. There is a lot not known about the reaction mechanism still. Chris and I have been working on something that's called a reciprocating subunit mechanism, and you'll probably hear more about that in the days to come and in person. And Chris has done a lot of cool molecular dynamics, but in conjunction with actually a former student that helped develop a lot of the MCC materials, we've developed a QMMM approach for looking at the reaction mechanism as well. So it's more than just molecular dynamics you can do depending upon the level of sophistication that you want to get into. And for the people that aren't quite sure what MMQM stands for, it's molecular mechanics, quantum mechanics, and it lets you look at bond breaking events and transition states, not just dynamics the way molecular dynamics. And then Jesse referred to the role of oligomeric structure, and we've got a whole bunch of cool things we've doing and done with that. So I think the last slide is a repeat of the one about the available things for the drug design, just to remind you that, there are a bunch of resources already out there and ideas about things. Again, they're all in the links. Definitely go to the CureNet page. As you're trying to develop a syllabus, CureNet is the best thing you can do, is go to the CureNet site, because Erin Dolan is a fantastic virtual mentor through CureNet. And if you visit our page, it's one of the approved exemplar pages on CureNet. And that's it, folks.
All right.
Thank you, Joe,
Excellent.
for
And just
saving my bacon there.
yeah, it's a team effort. And that's excellent because we have coming up now a break. So take a few minutes off of Zoom and do those things you need to do. Any questions quickly before we take our break?
I had a question about this one because I've been around the MCC community for a while, and I was just wondering what I feel like I have a little bit less of a grasp of what this cure might look like. Obviously, there's, different options, but maybe Ellis, could you give an example of some hypotheses that students have come up with for a general chemistry
Sure. So
yeah.
in most of the drug design cures that we've developed and used, the goal is to develop an inhibitor or a potential inhibitor that can distinguish between the human isoform and the pathogen isoform. And so I don't know whether you've heard of the term cryptosteric inhibitors, but we've developed this and presented this at a couple of meetings now, and it's been picked up on. But the idea of developing a cryptosteric inhibitor and then optimizing the structure of a cryptosteric inhibitor to try and distinguish between the host and the pathogen. So, they usually come up, we usually start, depending upon what level, we usually start with, for instance, this came up when we were having one of our sessions just last week with a friend of mine at UT Southwestern. You can start by saying, okay, are there folklore remedies, for instance, that might apply to a particular, and do any components in that inhibit malate dehydrogenase, and do they show specificity? So, that's one sort of approach. And then you get into sort of finding a cocktail that inhibits, and then you try and work out which part actually is the inhibitor. Or you can start by saying, okay, and I don't know whether you're familiar with Carla Matos and Dagmar Ringer's work on what is a binding site, but they developed a really cool approach to look at what sort of moieties bind to various pockets on a protein. And it's called multiple solvent crystal structure analysis. And so we use a computational version of that to look at where fragments of drug-like molecules might bind, and then we either grab the fragment and see if it does inhibit, or we try and find a friendly organic chemist, and this is why it's great for a collaboration with an organic chemist, to make a molecule that has two or more of those elements in, and we then say, does that now inhibit, and does it give us specificity? So, there's lots of different ways you can develop a hypothesis. Focus is always on the student developing the hypothesis. If I develop a hypothesis and tell them what to do, I've taken away most of the aspects that make it a cure. So, I want them to explore, look at the structure, look at, the big picture goals, and develop their hypothesis from preliminary data.
Thank you.
All right, excellent. Great questions. So a couple things that I was going to say. I want to take a break. I'm also going to ask if the people who presented could throw their slides onto the Cohort 26 channel, and then you can have references and links and so forth, access to that. And I'm also going to stop recording.